All right, here we are, looking at muscle physiology and how a muscle contracts. This is one of the more complex physiologies, especially for what we've experienced so far in AMP1. But here we go. Let's remind ourselves that we're talking about muscle. That's a specific tissue type. Recall from the connective tissue chapter, we talked about other types of tissue, and muscle tissue is one of them. We will be looking at specifically skeletal muscle in this chapter. We will barely touch on cardiac and smooth. We will save those two for later chapters, specifically the cardiovascular system. But remember, there are three types: skeletal, cardiac, and smooth. One of the ways to distinguish between them is not just the physical aspects that are different, but two of the three are controlled without you knowing. That would be involuntary. And then skeletal muscle can be both involuntary and voluntary. So if I want to make my arm move, I just make it move. If I want my heart to beat faster, I can't really say, beat faster. It won't listen. Same with smooth muscle. I can't say to my small intestines, contract or relax. They are controlled without cognitive input. In other words, any voluntary nervous system connection will not be made with those types of smooth muscle and cardiac muscle. So, moving on to what do they do? What does skeletal muscle do for us? Well, I would imagine that you would get that it allows us to move, right? So the muscle is what produces the force to move the body or move a part of the body. The bones are what allow movement, but skeletal muscle is what causes movement. And that has a lot of implications, right? It allows us to be mobile. It helps us to keep a certain position when we're sitting in a chair or standing up. But probably one of these that you may not have guessed is the maintenance of body temperature. And as soon as I say it, you're like, oh yeah, I get that. When you're really cold, you shiver. And the shivering is really a rapid contracting and relaxation cycle of the muscle so that it generates heat, right? Anytime you generate force, one of the byproducts is heat. So this is one of the provisions that skeletal muscle has for us. Guard entrances and exits. That's interesting, right? We'll get more of that when we do the digestive system and the urinary system. I'm sure you can imagine what that means, right? It's the sphincters, the not just yet control, right? Not ready to go yet. And then supporting soft tissues. You realize with your study of the gross anatomy of the bones and muscles that the abdominal organs are not protected by and large by bony structures. It's mostly the abdominal muscles that protect the underlying tissues. So they do that too. Now, some physics. Aren't you excited? Well, in order to cause movement, a muscle must cross the joint. Let's look at this example over here. Let's get my little pen here. See, here's the biceps muscle. And you notice that it starts around the head of the humerus, and the muscle continues along, crosses the elbow joint. Notice that it goes past the elbow joint and attaches to the radius, specifically the radial tuberosity. That's right here. See that? So just from appearance, you notice that the muscle starts here and ends here and crosses the elbow joint. That means when this muscle contracts, it will get shorter. That's what happens when a muscle contracts, and pulls on one end. It can pull on the radius, which will move the arm at this fulcrum. So we're going to be dealing with some of the common terms used to describe levers. Now, you've heard of levers before. Probably not in the context of the human body, but the human body is made up of 3 types of levers. Levers are just arrangements of 3 things: a resistance, the thing that you're trying to move; a fulcrum, a point in which the resistance pivots; and then an energy source, a source that will provide the force. So you're familiar with a wheelbarrow, right? You load the rocks in the barrel, and then you lift up on the one end and push. But where is the movement caused? That's at the wheel, right? So the wheel would be the fulcrum. The resistance would be whatever you put inside the barrel. And the energy source would be you. So if you look at these different classes of levers, we have first class, second class, and third class levers. All they are, the difference between the 3 are where each of these 3 components are in relation to the other. So in the first class lever, we have the energy on one side. Let's say it's pushing down. Then you have the fulcrum in the middle, and the resistance is being moved up. So a great example of that is the head. So the energy source, let's say it's this muscle here, the splenius capitis, is pulling down, right, down on its connection. And the fulcrum is the occipito-atlas joint. The head is the resistance. So you're trying to move the head up by pulling down on the back of the head at a fulcrum of the occipito-atlas joint, right? So does that make sense? So think about that. Pulling down the back of the head, it's moving here, and it's lifting up the head. How is that different on this side? Take a second to think about that. The fulcrum is down here at your toe joints, right? Your metatarsophalangeal joints. So that's where you're going to pivot at. The energy to move is going to be provided by your gastrocnemius muscles. They're going to contract and lift up on the heel. So the heel is lifting up. Now what are you trying to lift up? Well, you're lifting up all the weight that's pressing down in the center of the foot. Right? That's the resistance. So the resistance is lifted up by lifting up on the calf muscle, right? The calf muscle lifts up at the fulcrum. What does that look like? That looks like a wheelbarrow. It is a wheelbarrow. Very similar. Now, the third class lever is a little funky. The third class lever, the resistance is the arm here, specifically the hand. The fulcrum is the elbow joint. And the energy provider, the force, is going to be the biceps muscle. Now look, this is a little awkward, right? You kind of lift up in between the fulcrum and the resistance. It's kind of weird, isn't it? It doesn't seem like something that would give you that much of a mechanical advantage. Well, it's not the best of the 3, right? Probably the best of the 3 is the first class lever. It gives you the most mechanical advantage. But they all give you some sort of mechanical advantage, which is interesting, right? So the arrangement of the muscles and the bones create an environment where we always have some sort of mechanical advantage because of the lever system that is being utilized. So what I would expect you to understand is the differences between a first, second, and third class lever. And if I gave you a labeled diagram, you could tell me which one is which. So practice that. Okay, now let's look at the muscle tissue itself. Again, we are focusing on skeletal muscle tissue. So for me, I like to see this with my eyes. It's dealing with layers of a skeletal muscle. These are the words, the terms that we will use: epi, mysium, perimysium, and endomysium. Now you notice the prefixes, epi, peri, endo. Right? You Greek and Latin word readers remember that these are very important to help us figure out where, with respect to the layer and its relationship to surrounding structures. So this helps us. So epi is around, right, the outside. Peri is in the middle, and endo is on the inside. Let's see what that looks like. So here is a whole muscle, right? So if I took your bicep muscle out of your arm, and we looked at it, we took a cross section of it, right? This is what we would see right here. This is a cross section of the entire muscle right here. So I want you to notice they do a pretty good job here of changing colors. You look like this yellow group of connective tissue surrounding the entire muscle. That would be called the epimysium, right? The epimysium or mysium. This is the connective tissue layer that surrounds the entire muscle, epi. Now you notice that there's groupings of fibers, right? These groupings of fibers are called fascicles. So all these little red circles that you see here from the cross section, they're grouped together in fascicles. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 fascicles in this particular whole muscle. Now each of these fascicles you see is surrounded by what looks like to be some blue connective tissue, right? That's just color-coded so we can see them better. What do you think the name of that particular connective tissue is called? Right, perimysium. Perimysium is the one surrounding each fascicle. So now, before we go to the endomysium, let's recap for a second. If I ask the question, what connective tissue layer surrounds a group of fascicles? This is a group of fascicles. What surrounds it? The epimysium, right. If I said, what connective tissue layer surrounds a single fascicle? You would say perimysium. Good. Now, let's go back to this picture for a second. So they actually pulled out one of the fascicles, right? Here's a fascicle. Now, if you look at the muscle fascicle, this is where it gets really interesting. So this is a fascicle, just one of them. Each of these little things right here is called a muscle fiber. A muscle fiber. So they slid out a muscle fiber here, didn't they, from the fascicle. Now a muscle fiber, you see that it's kind of a pink connective tissue layer surrounding that muscle fiber. That's called the endomysium. Now this gets really interesting. The endomysium is connective tissue, and it's surrounding the muscle fiber. Another name for the muscle fiber is the muscle cell. This is the actual cell of the muscle. And you notice that a whole muscle is made up, right? This whole muscle is made up of groups, many, many groups of muscle cells. So let's take a second and see how you do with this. Okay. A group of muscle cells makes a blank. A group of muscle cells makes a blank. The answer is a fascicle. Right? Groups of muscle cells makes a fascicle. Groups of fascicles makes a blank. That's right, a whole skeletal muscle. So skeletal muscle is made up of groups of fascicles, which are made up of groups of muscle cells or muscle fibers. So let's even get more specific. A whole muscle is surrounded by an epimysium, and the whole muscle is made up of groups of fascicles, which are surrounded by perimysium. Fascicles are made up of groups of muscle fibers or muscle cells, which are surrounded by endomysium. Everybody okay so far? It's getting really interesting, right? So, okay, now if we take one of these guys out of here, if we want to see what a muscle cell looks like, this is a muscle cell. Now you notice it's got more red dots inside of it. So that means a muscle cell is made up of something. Well, it's a cell. It's got a nucleus right here, right? I'm sorry, right here. It's got a bunch of nuclei. It's got organelles like mitochondria. It's even got endoplasmic reticulum, which we'll call sarcoplasmic reticulum. And it's got all these red dots. Right? These red dots are called myofibrils. Myofibrils. Now this is a cell, right? It says muscle fiber, but we could also call it a cell. I'll try to write that with this. Probably not too pretty. This is also a muscle cell. Now a cell has a membrane, right? A phospholipid bilayer that separates it from the external environment. This one has one too. It's called the sarcolemma. Right, that's this thing right here. The sarcolemma is not connective tissue. It's a phospholipid bilayer. This is the cell membrane of the muscle cell. Right, so a muscle cell has a cell membrane called the sarcolemma. And it has connective tissue surrounding it called endomysium. Within the muscle cell are organelles that are important in, this is where the muscle contraction happens. It's an interaction of these myofibrils. So let's take a closer look at the muscle cell, right? So look, here it is. Here's some nuclei surrounded by a phospholipid bilayer called the sarcolemma. Now, if we look at the myofibrils, we see this is a myofibril. This is a single myofibril. So we're getting really, really close. We're zooming in. We see that there is a phospholipid bilayer that surrounds the cell, called the sarcolemma. Inside is going to be cytoplasm and tightly packed myofibrils. That's these guys right here. Myofibrils. These are the protein filaments where contraction occurs. So this is where we get a shortening of the muscle, where contraction or force is generated. Now you notice that there's a lot of stuff surrounding each myofibril. You got this blue stuff here called sarcoplasmic reticulum. That's really important. That's storing calcium, which we'll see later is extremely important in a muscle contraction. They got these yellow things that look like indentations of the cell membrane. So imagine you're traveling along the membrane and you're walking and all of a sudden, bloop, you fall down this hole. This hole would lead you inside of the cell through a T-tubule. So we have a T-tubule, an indentation of the cell membrane, which brings you inside the cell. That's right next to this blue stuff called the sarcoplasmic reticulum, which has what in it? That's right, calcium. Now there's a special region of the sarcoplasmic reticulum right next to the T-tubule here. That's this area right here, right? All the way, this one right here too, and all the way. Anything that's close to the T-tubules is going to be the terminal cisternae. So the terminal cisternae is a region of the sarcoplasmic reticulum that's close to the T-tubule. So you're kind of figuring out here that each myofibril is surrounded by a sarcoplasmic reticulum that contains calcium. That's going to be significant. Also, look at all these little purple things here, these mitochondria. Do you remember what mitochondria are for? They produce lots and lots of what? ATP, that's right. So they're going to be providing energy for this whole contraction process to happen. So you kind of get your bearings. This is a muscle cell, so it's a regular cell, has a cell membrane called the sarcolemma. This one happens to have particular unique features of these indentations, these T-tubules, that can bring something into the cell, mainly electricity, we'll see later. And right next to those T-tubules are a special portion of the sarcoplasmic reticulum called the terminal cisternae. And we know that this is where a lot of the calcium is stored. We also have some mitochondria here that will provide some ATP. So you have your bearings. It's really important. So you're getting really microscopic here on this myofibril, right? So you could say, are you ready? Groups of myofibrils makes a blank. A muscle cell or muscle fiber. Good. Groups of muscle fibers make up. Groups of muscle cells or fibers makes a fascicle. Groups of fascicles makes a whole muscle. Okay. All right. So how many myofibrils do we have? Well, each cell contains about 100 to 1,000 myofibrils per cell. That's impressive, right? Well, unfortunately, that's not the smallest thing we can deal with. Inside of the myofibril, we saw this. Let's back up for a second. Remember I said these myofibrils right here? These are made up of small filaments, myofilaments. That's the purple and red lines here. You notice how they're specifically arranged. That's not just an artistic rendition. That is actually pretty close to the way they look. So these myofilaments are what makes up myofibrils. It just keeps getting better, doesn't it? These myofilaments have a name. The thick one's called myosin, and the thin one's called actin. So the two main filaments inside of a myofibril, which is inside of a muscle cell, are actin and myosin. There's another myofilament called titin, and it provides the elastic component of muscle. We'll be mainly talking about actin and myosin. Just keep titin in the background. Now, how this actin and myosin arrange is pretty cool. Let's look here. Here's an example of actin and myosin and their arrangement. You see the purple line, the thick purple line with little bumps on it? That's the myosin. So these are strands of myosin. And the little thin red lines are the actin. You notice that they're arranged a certain way where the actin surrounds the myosin. We'll see that better when we look at a cross-section view. The first one we're going to look at is actin, the thin one. Now, it's not a red line. It actually is a protein filament. It means it has a combination of protein arrangements that make it up. This is what it looks like zoomed in slightly on the red line. So you see it's kind of twisted around each other. Then we zoom in even more and we see, okay, now this is getting real interesting. So right now we're labeling the parts of actin, which is a myofilament that makes up the myofibrils that are in the muscle cell. Keep doing that. Okay, so what are the parts of actin? Well, they got these little balls here. These little balls are called the F-actin strand, made up of G-actin molecules. So the little balls are G-actin molecules that are arranged in a strand called the F-actin strand. So they're kind of twisting, almost like an alpha helix, like DNA. But that's not all. It looks like these balls have little yellow spots on them. Can you see the yellow spots right here? Each ball has a little yellow spot. Now, it's not yellow inside your body, but this is just for the illustration. There's a region on the G-actin molecule that's different. We call that area the active site. That particular area attracts myosin. Remember, the myosin and actin are close to each other. So there's a part of this G-actin molecule called the active site that attracts myosin. But you notice here that it's covered. It's covered by this strand of protein, this green one here. This is called Anyone know? Tropomyosin. That's right. Tropomyosin is the strand that covers the active site, so myosin can't see it. Now, on top of this tropomyosin are these little brown strands here. They're called troponin. So troponin is connected to tropomyosin, which covers the active site on actin, right, on the F-actin strand. So you got that? So what covers the active site on the G-actin molecule? Tropomyosin, that's right. And what is connected to the tropomyosin? Troponin. Now you can file that in the back of your head because we're going to talk about these when we talk about how do we get actin and myosin to interact. You can probably start to think about that now. So let's take a look at myosin. Myosin looks very different. It's thick. It's much, much thicker. And it's made up of multiple strands, protein strands, that have a long tail and a big head. So you put a bunch of these things right here, this myosin molecule together, and you get a strand of myosin. You notice that these heads are all the way around the strand, and they're located in a high frequency, right? A lot of them next to each other. Now, the interesting part is this head is movable. It can reach up, and it can move and pull. Now, that's going to be significant, right? Because this myosin is going to need to reach up and grab the actin and move it. That's how the muscle is going to contract. So if you remember the parts, we'll see them again later when we look at the whole process in one of your interactive physiology videos. Okay, so how are these arranged, these actin and myosin? Well, they're arranged in what's called a sarcomere. Right, that's this over here. The sarcomere, that's right here, is the functional unit of muscle. It's how the actin and myosin are arranged. You notice that there's a line here that looks like a Z, this red one. That's called the Z line. So from this Z line here to this Z line here is called a sarcomere. So from this Z line here to the next one, that's another sarcomere. So you notice that the sarcomeres are arranged one after the other. That's called, they're arranged in series, one behind the other. And that means the length of this muscle cell is going to have lots of sarcomeres one after the other in series. Now, if you look at just one sarcomere, right, this thing blocked off here, it's got other parts that help us tell what the interaction between actin and myosin looks like. In the H zone here, this first one, what do you see? Just the purple line. So what would you say is in the H zone? That's right, myosin. Now, the next one we have is the I band, which goes from the end of the myosin here all the way over to the end of the myosin here. Now, do we see any actin in this A band? Yes, we do. Do we see any myosin? Yes, we do. Now, within this A band, there's a point at which the actin and myosin are overlapping each other. And guess what we call that? The zone of overlap. That's nice, isn't it? Okay. Next, we have the I band. Now, this particular region goes from one sarcomere, end of the myosin, to another sarcomere, right? So this is sarcomere 1 over here, and this is sarcomere 2. You notice that this particular landmark or region spans 2 sarcomeres. But what do you see? You just see the red, right? The red lines. That's the actin. So in the I band, you see actin. In the H zone, you see just myosin. And in the A band, you have a region where they overlap, called the zone of overlap. Now, the green little line here is called titin, right? We said that that doesn't necessarily play a role in the contraction process directly. It's an indirect role. It helps bring this stuff back to its resting state. This is an actual micrograph of a sarcomere. This is the real deal Holyfield down here. You got the Z line. You got the M line down the middle. You got the H zone. You got the zone of overlap right here. And then you have just actin here. That's pretty cool. Okay, so functional unit of skeletal muscle, sarcomere. How is it arranged? You remember to explain and recognize and be able to label one of these puppies. And tell me what's going on. If I said, what would you expect to find in the H zone? You could say, the anchoring point of the myosin, which is the end line, and myosin. What would you expect to find in the I band? Primarily actin with some titin, and the Z line. What would you expect to find in the A band? And you can say, well, on the region of the zone of overlap, you would expect to find actin and myosin both. Okay. Now, remember I said we would like to look at this from a cross-section view. This is kind of neat. So if you took a section and you cut down the length of the zone of overlap, this is what you would see. This is pretty cool. So remember I said that actin and myosin interact to cause a contraction. Well, look at the arrangement. Let's get this pen going here. See, the red dot's the actin, right? You're surrounded by 3 myosin. So you got 3 myosin that can pull on this one actin. Now, not by itself, right? This myosin is going to pull on this one and this one. And so we got to figure out what's the relationship between myosin and actin. Well, look, for every myosin, there's how many actins surrounding it? 6. So this is a highly interacted region between actin and myosin for a specific reason, which we'll get to. Okay, so let's recap. Whole skeletal muscle surrounded by what? Epimysium. Groups of muscle fibers called fascicles are surrounded by what? Paramecium. So this is a fascicle surrounded by paramecium. This fascicle is made up of groups of, what are these things? Whoops. What are these right here? These are muscle cells. Good job. So a muscle cell or a muscle fiber surrounded by what connective tissue? That's right, the endomysium. But what's this here? What's this smooth pink membrane? That's right, it's the cell membrane, the sarcolemma. And then inside the cell is where you got all the goodies, right? The myofibrils, which are surrounded by sarcoplasmic reticulum and T-tubules from the membrane. And these myofibrils are made up of myofilaments that are arranged in sarcomeres. The myofilaments, the 2 main ones are actin and myosin. Good googly mugs. Okay. All right. Let's take a pause here. We're going to go and discuss some background information that you do not have in this chapter. I repeat, you will not find the following information in the chapter. This is to help you understand how a muscle contraction works. We have to understand the cells, the life of a cell. What is the electrical and chemical environment of a cell that makes it able to respond to changes? That's our goal here. We're going to define what the transmembrane potential is. That's going to take some time. So remember, this is not something you will find in Chapter 10. This is taken from the first nervous system chapter and from Chapter 3. So it's some stuff you've heard already, but also some new terminology that's very important. So we're going to take some time to kind of build this foundation so that we can move on and discuss how a muscle contracts. All right. Now we're going to remind ourselves what a cell looks like. Remember, a cell is surrounded by a phospholipid bilayer, which is hydrophobic heads on the outside and inside, and, did I say that right? Hydrophilic heads on the outside and inside, and hydrophobic tails in the middle. So remind me, if I was an ion, a sodium ion or a potassium ion, which is a charged particle, right? Would I be able to pass through the phospholipid bilayer? The answer is no. So what a phospholipid bilayer does is controls the permeability of a cell. Now, where it gets interesting is the fact that, for the most part, you've learned that particles, if they're allowed to move across a membrane, will move until they reach equilibrium. But the cell is much more complex. For one, it doesn't let everything pass through it, so it's selectively permeable. And in this case, we will learn that we can alter the permeability of the phospholipid bilayer by using channels that pass through the phospholipid bilayer. And we will see that these channels can have a profound effect on what's inside the cell and what's on the outside of the cell. So much so that in this case, they will not reach equilibrium. They will not be in equilibrium. So we're going to have to wrestle through all of these new applications of information that we already understand. We are very familiar with the phospholipid bilayer. We are very familiar with protein channels. We are familiar with ions, specifically the sodium and potassium. We're just going to now apply that knowledge in how a cell membrane looks like at rest when it's not being asked to do anything except sustain life, right? There's no change in the environment. Okay, that's our mission. Okay, so let's take a look. First thing we have to realize is that there are channels on cell membranes. It's not just a phospholipid bilayer. We know that. And there are two classes of channels that we're going to discuss. A passive channel, which means that it's open all the time. I call it the diner, 24/7. Now, these channels don't let anything pass through. They're very specific. So you'll have a specific channel for a specific component that it wants to let pass through the cell membrane. But anything else that comes along will not be able to pass through that particular channel. You need a different channel. Then we have active channels. These are gated channels. They need some sort of stimulus to open them up. And we'll see examples of both passive and active channels. Okay. Now, how do things move across the membrane? Well, you already know this one, right? You know that you need a concentration difference in order for particles to move from one side to the other of the membrane. We did that with diffusion and osmosis, right? We said that there must be a concentration difference. Now, we're not talking about moving water here. We're talking about moving ions. So that means we have to have more of one ion on one side than we do on the other. There are lots of ions inside and outside of a cell. We're going to be dealing primarily with sodium, Na+, and potassium, K+. Chloride, Cl, negative, is also one that we might see here and there. But the two major ones are sodium, Na+, and potassium, K+. So in order for sodium to move from one side of the membrane to the other, there must be a difference in concentration. There must be more sodium on one side than there is on the other. In order for potassium to move, the same is true, right? So if we're looking at the amount of the particular compound on one side compared to the other, we're talking about a concentration difference. But that's not the only driving force to move something across the membrane. Notice and remember that these particular compounds have, or I should say elements, have charges. That's why they're ions. So it's not just Na, it's Na+. And it's not just K for potassium, it's K+. So these have charges. So that means if there's more positives on one side of the membrane and more negative or less positives on the other side, that's going to create a gradient too. That's called an electrical gradient. So it becomes interesting because sodium and potassium both have a chemical gradient and electrical gradient. So we call that a chemoelectrical gradient or a chemical electrical gradient. So this is what drives the forces that move these particles across the membrane. So the energy is found in the difference in concentration of electrical gradients or chemical gradients. Now, that's not the only way to move things across the membrane. We're getting even more complicated, aren't we? Well, those that we just discussed were passive forces, meaning that the cell doesn't have to provide any energy because the energy is found in the gradient. But let's say you wanted to move something against its gradient. Remember we talked about this? Imagine if you're in a canoe going downstream, so you're going with the current. You don't have to paddle at all, right? That's the passive movement. But if you decide you want to go back to where you came from, so you turn around in the canoe, you don't just automatically start moving the other direction. You now have to apply a force. You have to paddle. And that is the example of active forces that move things across the membrane. So this requires our cells to use up ATP, its energy source. It has to provide the energy. So that means that these ions are moving against their electrochemical gradients. They're going the opposite direction. And you're saying to yourself, why would you ever want to do that? Why would you ever want to paddle upstream? Well, let's keep this in mind, that these are two ways in which things move. And then we'll see what the significance of each of them are. The prime example of an active transport is the sodium-potassium ATPase pump. This particular pump pumps sodium against its concentration gradient and potassium against its concentration gradient. So we'll have to see this one in action too. Now here's a picture of all of it working together. I don't expect that you understand everything you see here. This is just a kind of a beginning picture of what's happening. Let's just take a second to kind of orient ourselves here. You notice that this is the phospholipid bilayer, the blue bar that's going across here. And these positive and negatively charged things can't just pass freely through. Now we have these channels. Now it looks like these channels, this purple one here, oh, that's a bad circle, but you get the idea. This purple one here lets the purple things pass through it. Now it looks like as long as there's more purples on the outside than there are on the inside, these purple things will move. So do you think this is an active or passive channel? That's right, this is a passive channel. All we needed was a difference in concentration. Or in this case, even a difference of charge, right? There's more negative on the inside. This is a positive charge. That will then come inside to balance the charge. Now what about this orange one over here? Well, it looks like this is letting this particle pass through this orange one. This one's going out. I wonder why it's going out. Well, this one came in because there was more of these purple circles on the outside than there are on the inside. So they moved in. Well, it looks like there's more of these orange circles on the inside than there is on the outside. So it moves out. These are both examples of passive movement through a protein channel that is specific to each. Could this orange one pass through here? No. Could this purple one pass through here? No. So these are two of the main leaky channels that you will find on a cell membrane, a potassium leaky channel and a sodium leaky channel. Now this thing in the middle, what the heck is this thing? Well, the first thing you notice is that it uses ATP. The second thing you notice is it's moving these orange particles inside the cell, which is where they're more concentrated. That's going against the concentration gradient, right? Because concentration gradients always move from blank to blank. That's right, high to low. So we're going against the concentration gradient to get these orange things back in here, therefore we need energy. Well, also, it seems to be moving these purple things from inside the cell to the outside. That's going against the concentration gradient of the purple things. So this sodium-potassium exchange pump uses energy to pump 3 sodium out and 2 potassium in. Yes, it is significant that there's more positives leaving than there are positives coming in. That's going to be something we're going to have to figure out soon enough. But you kind of get an idea, a visual of what's happening here. Okay, so we've established that there's going to be an electrochemical gradient, meaning that in this picture back here, we saw that there are more purples on the outside. We could say that these purple things are sodium, right? So the concentration of sodium ions is greater on the outside of the cell than it is on the inside of the cell. We could also say that the concentration of potassium is greater on the inside of the cell than it is on the outside of the cell. Now, does that have significance? Well, we'll have to check it out and see. Okay, so let's get some things straight. Potassium leaky channels. They allow potassium to go from the inside of the cell to the outside of the cell. Why that direction? Because potassium is greater concentration where? On the inside. So there's a concentration gradient that allows potassium to leave. So the net movement of potassium is out. What about sodium? Just the opposite, right? Sodium is higher on the outside than it is on the inside. Therefore, sodium is going to come in through their leaky channels. So this gets really interesting. We're moving sodium in and potassium out. Now, remember what I said before. You have always learned that if there was a leaky channel and there's a concentration difference, whatever is moving through that channel would move until equilibrium is reached. And I said that is true, except in this case. So we're going to have to figure out what the dealio is here. Let's see if we can find a good picture. I think I have a good picture here somewhere. Don't get too crazy. We'll come back. Maybe not. I thought I had a good picture, but I don't. So let me go back to this one picture here. Now, what if, since I can draw somewhat, what if I put another potassium leaky channel here? Well, maybe I'm not a great drawer, but you get the idea, right? Hopefully. This is another potassium leaky channel. So this one and this one are the same. Let's put another potassium channel here. So I'll put a K, just so we know it's potassium. It's really hard to do. And this is a K. So what would your observation be as scientists here? So if you're looking at this cell membrane, you would say that there are more potassium leaky channels than there are sodium leaky channels. So let's say that there is more of this that's going to then escape, right? Right, and so what's the significance of that? What's the fact that more potassium would leave the cell than sodium would come in? That's right. More positives would be leaving the cell than more positives coming in. Is that a balance? No. That means the inside of the cell would be more what? Negative, that's right. And look, this is showing that, more negative on the inside. So that is the way a cell membrane looks. There's more potassium leaky channels than there are sodium leaky channels. Therefore, more positive ions leave than come in. Now, how do we control how negative the inside is? Because this seems like it would keep going until equilibrium is reached, right? Well, we got this thing. And what do we say this does? This pumps 3 positives out and 2 positives in. So that means this is pumping more positives out than it is in. So what's that mean? The inside is going to be more negative. That's right. In fact, it's going to be about negative 70 millivolts. So this is a voltage meter. And it says between the fact that we have more potassium leaky channels, allowing more passive potassium leakage than sodium leakage, right? More sodium, less sodium is coming in than potassium is leaving. And we have the sodium-potassium pump pumping more positives out than it is in. Those two things together are going to provide this negative 70 millivolt resting membrane potential. That means we're not at equilibrium, right? There's more positives on the outside than there are on the inside. That creates what kind of gradient? That's right, electrical gradient. And we also said we have more sodium on the outside than we do on the inside, and more potassium on the inside than we do on the outside. That creates a chemical gradient. So we have a chemical gradient and electrical gradient at rest when the cell is relaxed. Now, why wouldn't we want it at zero? Right? Why wouldn't we want equilibrium? That is the brain cramper. Well, we don't want equilibrium because we want to be able to respond to changes. If everything is balanced, there's no need to change, right? There's no way to respond to a change around the cell. If we keep it out of balance, then we can respond to changes. This is what makes our cells excitable. The resting membrane potential is what makes our cells excitable, right? These are just the words. You can make a story with a picture, but you have to have words also, right? You have to be able to explain it to someone. These are the words I just said. I said, because the plasma membrane is highly permeable to potassium ions, right? More leaky channels, potassium leaky channels than sodium leaky channels. You get a resting potential of negative 70 to negative 90 millivolts, right? Because there's less sodium leaving, this has a very small effect. So the main contributor thus far to the resting potential is the fact that we have more potassium channels. But also, what maintains that difference is the sodium-potassium pump, right? Because it pumps 3 sodiums out and 2 potassium ins. That means there's more positives leaving than there are coming in, keeping it negative 70. That's an average resting membrane potential. Okay, so we've established the basics of a cell membrane. We've said that there are channels that help determine the membrane's condition. We said specifically potassium and sodium channels, and they're going to contribute to the resting potential. Now you're going to have to get very specific. Go back and listen again if you have to. But you really have to understand what the resting membrane potential is and how is it established. Okay? Where it is, what it is, and why it is. Always what you have to do when you're given a new concept. And this one is no exception. Okay. Now, because we have this resting membrane potential, we can respond to changes. Right? That's the whole point. We can respond to changes. Now, what kind of changes? Well, there are chemical changes, there are electrical changes, and there are mechanical changes. Right? When I touch my skin on my hand, how do I know that I can close my eyes and know that I'm touching my skin, right? How do I know that? Because there's a mechanical change in the cell membranes that I'm touching. And there's a way to communicate that to my nervous system. That's pretty interesting, right? You know, how do I smell grandmom's apple pie? Because there are chemicals that attach themselves to the cell membranes of receptors in my nose that change the permeability of those cells so they can communicate to my brain, says, oh, that's grandmom's apple pie. How do I get a muscle to contract? I change the electrical current across that cell membrane, which will change the permeability. You get the point here. There are multiple ways to change transmembrane potential. So that means we must have different kinds of channels. Well, let's see. There are. And guess what the 3 classes are? Chemically gated channels, voltage or electrically gated channels, and mechanically gated channels. So let's look at them individually. Chemically gated channels. Guess what they respond for? Now remember, this is the solid blue bar that represents the phospholipid bilayer. And guess what it's got on it? Potassium leaky channels, sodium leaky channels, the sodium-potassium pump. They're all here. You just don't see them. So they're maintaining this negative 70, this more negative on the inside than it is on the outside, at rest. Now some chemical comes along, the red dot, and it binds to this chemically regulated channel. And guess what happens to the channel? It opens up. Now what happens? Well, you've established a gradient. Now the ions can move into the cell. What does that do? Well, this is a positively charged ion. It's now going to change the inside of the cell, therefore the membrane charge. That communicates something, right? There's a change. Would these positively charged ions move if they were equilibrated on both sides? If there was 3 on the outside and 3 on the inside? No. So because we have a resting membrane potential that makes a difference in concentration, when there is a change, we can respond. In this case, when there's a chemical present that opens this channel, we can now change the electrical state of the membrane. So what do you think the next one is? Voltage. Well, what do you think is keeping this channel closed? A certain voltage. If you change that voltage, well, it doesn't look like it changed much, but it did here. It went from that little bit of space to so the voltage went up. This channel opened. Now what's going to happen? More positive ions are going to come in. Does that change the voltage? Certainly does. Look at that. Change the voltage big time. And when you get to a certain voltage, that'll close the channel. So how are these channels regulated? By the change in voltage. Good. The next one. These are mechanically regulated channels. When you change the physical arrangement of the cell, like pressing on your skin, right? That cell membrane gets distorted. These channels open up and change permeability, right? And can now communicate. So, 3 main channels that help us respond to a changing environment. They are chemically regulated or gated channels, voltage gated channels, and mechanically gated channels. Good. So, now, because we have established a resting membrane potential that gives us a gradient, and because we have channels that can change permeability and allow those ions to pass through, we can communicate change. Now, the change in the voltage of this plasma membrane, we said at rest it's about negative 70, right? And any of those channels, the voltage, the chemical, the mechanical, can change that number, that negative 70. If they let positively charged ions come into the cell, that negative 70 is going to be more positive or more negative. That's right, it's going to be more positive. We call that a depolarization. So, the way that we influence a cell membrane is by these graded potentials or local potentials. The names for them are depolarization and repolarization. What do we start off at? Rest. Good. Negative 70, right? So in this case, there was a change in the voltage. We brought more sodiums into the cell somehow. Just a little bit. Maybe the cell next to it changed its sodium on the inside and the sodium came over to this next cell. And now this becomes a little bit more positive on the inside. These gates are going to do what? Open because they're voltage regulated. Now sodium really comes in, changing the membrane all the way to plus 10, right? We got serious change. Now, can we stay there? Can we stay at plus 10? No, that's right. This is communicating a change. We've communicated the change. Now what do we have to do? We have to get back to rest. How do we do that? Well, we open up a channel that will let positive ions leave. And which ones leave? Potassium. Good. So at a certain voltage, this plus 10, the sodium one's going to close and the potassium is going to open, letting the orange ones out. What's that going to do to this number? Make it really negative. In fact, it gets to about negative 90. Is that where we are at rest? No. We're at negative 70. What's going to get it to negative 70? The sodium-potassium pump is what maintains this negative 70. So we'll go back to rest until what? Another stimulus comes in, another change. And we'll keep doing this as long as there's a change. When you stop the change, this is where we stay. Now, I know this is complicated, but you do have to remember terms like depolarization and repolarization and resting potential, and the fact that it's sodium and potassium ions that we're moving. These are key concepts to understand a muscle contraction. And we will again revisit this in the nervous system. It's that important. And all other systems. So this is something that you're going to have to spend some quality time trying to understand. So here is a graphical representation of what I just said. This is rest, right? The blue. We're starting at negative 70. Some kind of graded stimulus comes in and changes that to a point where we cause the permeability to change drastically. We call that a threshold stimulus. We reach that threshold of stimulus, we're going to really change the membrane's permeability, right? We're really going to change it. We're going to go from what was originally rest, we're going to zoom up to plus 30 in this case, right? And now if we go from a negative number to a positive number, we call that depolarization. So as a result of this stimulus, this threshold stimulus, we are now depolarizing the membrane, making it more what? More positive, good. Now once we reach to a certain peak, then we're going to start to come back down, losing the charge, becoming more negative on the inside. That's repolarization, right? We come down here to about negative 90. We get back up to our rest again through the sodium-potassium pump. Okay? So that's kind of a cycle. The reason it changed is because there was a stimulus that came in, right? Some sort of stimulus. We'll see what that looks like with the muscle. Okay, it's a lot of information, a lot of terms. Make sure you take your time. You stop when you have to. Think about what you're hearing and reading and making your own story. Okay, so this is a kind of a recap. You can read the resting state, depolarization, repolarization. This term hyperpolarization means we go below the negative 70. And how do we get back to negative 70? Sodium-potassium pump. Good, good, good.